This week in Pediatric Oncology, the podcast exploring hot topics and exciting advances in childhood cancer. TWIPO is produced by Solving Kids Cancer, nonprofits located in New York and London dedicated to improving research and supporting families, because every kid deserves to grow up. Subscribe to TWIPO through your favorite podcast platform.
This week in Pediatric Oncology, the podcast about new advances for childhood cancer. Hi, everyone. Welcome to episode 113, recorded on December 20th, 2024. I'm your co-host, Brenda Weigel from the University of Minnesota in Minneapolis. I'm here along with my co-host, Dr. Tim Cripe. Tim?
Hey, Brenda, good to see you again and looking forward to this.
And Tim is from Nationwide Children's Hospital in Columbus, Ohio, affiliated with The Ohio State University. Today on This Week in Pediatric Oncology, we have a wonderful guest, Dr. Nicole Drezner. Dr. Drezner is a pediatric oncologist and the Deputy Division Director of the Division of Oncology 2, also known as DO2, of the U.S. Food and Drug Administration, also known as the FDA. Dr. Drezner is a trained pediatric oncologist, and she completed her fellowship in pediatric oncology at Nationwide Children's Hospital, or at Children's National Hospital, I apologize, at Children's National Hospital, where she also completed an additional fellowship in pediatric neuro-oncology before joining the FDA in 2016. And so we are thrilled to have Dr. Drezner here to talk to us a little bit about what it's like to be a pediatric oncologist at the FDA, and also eventually to talk to us about some of the exciting work that has come out of the FDA, specifically in neuroblastoma recently, and a recent publication in the Journal of Clinical Oncology. Dr. Drezner, welcome. We're thrilled to have you. And tell us a little bit about what it's like to go from being sort of a practicing pediatric oncologist, a neuro-oncologist at that, to working at the FDA, and what your kind of roles, responsibilities, day-to-day work is like.
Thanks so much, Brenda and Tim. I'm really thrilled to be here and to share a little bit about the work that we do at the FDA in pediatric oncology. So as you said, I'm a pediatric oncologist and also did a year of neuro-oncology training at Children's National. I actually, as a child, wanted to be a pediatric neurosurgeon, and through a school project, met a pediatric neurosurgeon named Fred Epstein, who was working at NYU at the time. And interviewed him for that. Sent a letter and interviewed him for that project. And then during that interview, he sort of offhandedly said if I was still interested in pediatric neurosurgery when I turned 16, I was in 7th grade, that I could volunteer, for him that summer. And so I remembered that when I turned 16, I sent him another letter and asked if he, wanted to make good on that promise, and he did. So I spent the summer volunteering. At that time, he was at Beth Israel in New York City. So I spent the summer volunteering in children's playroom and it was a neuro-oncology floor. And that was my first direct experience with children with brain tumors. And I loved it so much. Realized in medical school and that I didn't really love being in the operating room. And so I sort of transferred over to the medical side of things and went for a pediatric residency and then a hem-onc fellowship and neuro-oncology. So really it's been a long-standing goal of mine to work with and impact children with cancer and children with brain tumors. I went to the FDA out of fellowship because I was at that time became very interested in how to sort of affect change, not just one-on-one with the patients, which is so vital, but sort of in a different way. And I really, I enjoy writing and reading, and that's a very large part of my job now. And so when I interviewed at the FDA, sort of offhandedly, I didn't know much about it. I really liked what they said that their sort of mission was and how they worked here. And I felt that it was very important in order to impact the field of pediatric oncology, that there are pediatric oncologists also working in the FDA alongside, the pediatric oncologists who treat patients every day. I think you really need somebody who knows the field in all areas of development in order to really make change happen. And so I think I transferred over sort of the love I had for treating, working with patients one-on-one to doing that same thing, at my job now, just from a sort of from a distance, but still with the same, general desire to improve the field of pediatric oncology and treat patients with cancer better and more effectively. So that's sort of how I ended up at the FDA as the deputy director for the Division of Oncology 2, or DO2, that oversees pediatric solid tumor development, rare tumors, and CNS cancers. My primary job is to oversee all aspects of development, from the first in human study to the new drug application or biologics licensing application for new products in any of those tumor types. We have a very dynamic and vibrant staff that is frequently meeting with sponsors, drug sponsors, advocacy groups, academic oncologists, and trying to build that into all of the advice that we give anyone who is doing research or developing drugs for pediatric oncology.
That's fantastic to hear that we have an advocate in the FDA. For most physicians, the FDA is a big black box and mysterious. So can you shed a little light on that, inside that box, for us? So you said it sounded like a lot of meetings.
Yes.
What is sort of the major, like from a pediatric standpoint, challenges? Do you have to educate people a lot in terms of what the differences are? What kind of hurdles do you run into? Tell us some more about that.
Yeah, so you're right. There are a lot of challenges, and I actually started out my career at the FDA on the thoracic oncology team, which is a much different space than pediatric oncology in general because of the number of patients affected and the drug companies that are developing therapies in lung cancer. So moving over into the pediatric oncology space, I think one of the first challenges that we are all, me and you and everybody who's involved in conducting trials for pediatric patients with cancer, is that it's thankfully a rare disease. There's small patient populations, but on the other side, that makes it very difficult to develop new therapies for patients with cancer, given the many challenges that there are in conducting trials in rarer patient populations. So the first thing that I saw when I sort of started out as the deputy director is that I really wanted for all of the people involved in pediatric oncology drug development to know who we are, and that we are also, all of my team are pediatric oncologists. And so we're sort of operating on that level. And we've done a lot of sort of forward facing outreach so that people know that we're real, pediatric oncologists with email addresses. We will show up to meetings. We want to be included. We want to partner, with anybody who's thinking of designing a trial in any way that we can, through through our formal meeting process, or even through informal meetings when that's appropriate. So that challenge, I think, is, I think that has, at least in the 2 years since I've been deputy director, I think that is improving. I think that you guys know more who we are, and are, and are willing to get in touch with us whenever there's something we can either help with or provide advice on. But I still think that, for pharmaceutical sponsors, I think that we still want there to be a little bit more, involvement of us in pediatric development, so that we can really help in the best ways, creating, seamless and efficient, study designs that will move the field forward to, everybody's advantage. So that's been the major challenge, just getting people to know who we are, that we are on the same, we're pediatric oncologists. We want to, want to do that work, and we're very reachable, approachable, and want to help.
And I think sort of, Nicole, we've certainly heard that, and really encouraging sort of early involvement of the key stakeholders in pediatric oncology, especially with members of the pharmaceutical or biotech industry. For our listeners, can you walk through just a little bit? You mentioned sort of the formal process and the informal process. And Tim said, a bit of a black box. Can you describe what sort of the general steps are and where, if you say early involvement, what does that mean? When would we want people to be encouraged to be involved?
Yes. So in the formal process, anyone who is thinking of, initiating a trial for any, pediatric oncology disease, the formal meeting process typically would start out with what's called a Type B pre-IND meeting, which is a formal submission to the FDA that describes in a meeting package there doesn't need to be a full protocol, but at least some details about what type of clinical protocol you are intending to develop, for what disease, what is the non-clinical or preclinical work that has been done for that drug, and sort of the reasons for, the elements of the protocol and the development program. We have different timelines for our formal meetings. So the pre-IND meetings are always designated as Type B, and there's types A, B, C, D, and then a few other types. But Type B, we have to respond to and schedule within 60 days. And in that meeting, we will go over our assessment of the strength of the preclinical data, any clinical pharmacology information on dosage that needs to be conveyed, and as well as our sort of overall both general and specific comments we have in a protocol. So that's usually where things start out. Just before an IND or before a research protocol, before a clinical trial is even initiated, we can provide advice on that. And it's especially important in pediatric oncology because very often one trial can be the only chance that we have to get data, just given the patient numbers. And so we would like, so we want to sort of have input as soon as possible, ideally before the trial has been started or even fully designed, so that it gives it the best chance of success at ultimately leading, to a labeling update or a new indication for that therapy. There's multiple meeting types, formal meeting types that can be then requested throughout the process. That generally fall under the Type C, which is sort of a general meeting type for any aspect of the drug development program that needs discussion, or a Type D meeting, which is if there's a specific question that's smaller in scope. And the types really just relate to the timelines that we have to respond to the meetings, with Type C being longer. But we expect, and we would like to have meetings, throughout the development process, through our formal process.
I think just pivoting a little bit of the conversation to the paper that you published on DFMO, eflornithine for neuroblastoma, that was recently approved. And I really appreciate your article because it brought a lot of statistics to the discussion and helped sort of make us feel, because there's been a little this has been a long thing. One of the things that's come out, right? The first trial was initiated in 2012. We're here now, 12 years later. How long it takes to get the data and to get an approval is one sort of issue, I guess. But since you're talking about early meetings and the best chance for success, it felt like, okay, they didn't do a contemporary randomized trial. You talk in that paper about how it is, one of the first that's approved with this external control and all that. Was that because they didn't come to you guys early enough, or because just the way these things evolve and they already had data that looked pretty good and, we need to do something with this data. Can you speak to that a little bit?
Yeah, so so you're right. The article did, sort of make those points that this was the first drug that was approved based with the primary, evidence being from an externally controlled trial. Yes, they did come to meet with us. The application was not submitted until November 2022, but we were having meetings as far back as 2018 and, even before about the development program. And I think it's more sort of the latter that you said. Tends to be sometimes the way things develop in pediatric oncology rather, where a single arm trial is done, particularly in a space where it would be difficult to, randomize. In this case, this was a maintenance setting for neuroblastoma when children are have completed upfront therapy. And so the standard of care, was to be done with therapy at that time. And so the sort of the way a randomized trial could have been conducted would have been, against a placebo. And typically that's not the way trials start. So this happens when we have a single arm trial, because that's considered sort of the most feasible way to get the drug to patients, and get early signal seeking, or early signals of activity. And then there is a signal of activity, and then it becomes much more difficult to then do sort of a randomized trial. And in this case, we thought that given the unique placement of this therapy in the neuroblastoma treatment paradigm, whereby it's given after all of the upfront, year and a half of upfront therapy, that and the fact that the results were known for the single arm trial, we thought our FDA's assessment it would be likely infeasible to conduct another large randomized trial, given that in this sort of unique circumstance, patients would need to be randomized to placebo because there was no other therapy to be given after the completion of the, upfront neuroblastoma therapy. So that was one of the earliest determinations that we had to make to even decide whether it was appropriate to rely on an external control. And then once that decision was made, the other sort of aspect was the effect size that was observed in the single arm trial large enough to overcome the uncertainty that is inherent in any externally controlled trial, and really is going to be there no matter how good the external control you use are. And so we had two things, unique circumstance, results known, difficult to do a randomized trial, plus large effect size seen in the single arm study, that we thought would have the potential to overcome the uncertainty in the externally controlled trial. So that's sort of how we decided, that yes, it is appropriate for us to consider this external control, for this drug application. And we also sort of had the benefit of there being a very high quality data set in the Children's Oncology Group AMBL 0032 study, that one had already been reviewed by us because it led to the approval of dinutuximab, and two, provided a very well matched control set, considering most of the patients, the vast majority, over 90% of the patients who received DFMO in the single arm trial had just come off AMBL 0032, and thus were the sort of same group of patients as were the rest of the 0032 patients.
And Nicole, that was sort of one of the things I wanted to ask you a little bit more about, was sort of the choice of the control data set, and is that something that the FDA, you as the FDA, decided on what is that appropriate data set? What elements are critical to that comparative data set? You've touched on a few of them, but but if you're comparing to that kind of external control, can you speak a little bit to what went into the decision making around selecting the control?
Yeah, so that's a great question, and it is one that we have published guidance about and sort of addressed a little bit in our paper as much as we could with the time constraint. But yeah, so in any in any trial, there are unknown unknowns, right? So there's things that may affect the activity of a drug that we don't know about, and therefore cannot control for. In a randomized trial, you assume that those fall, relatively equivalently between the two arms, and so the factors will be generally matched. But in external control, you obviously can't do that because you're not randomizing to the control arm. You're selecting a control that you are hoping will be as similar as possible to the experimental arm, in this case, the DFMO single arm study. So what some of the things, Brenda, that you said that we would look at or would want, for any external control, is that one, we need to have patient level data. If this is going to be the primary support of effectiveness for a new drug application, it would be extremely difficult for us to rely on any kind of data that for which we don't have the patient level data set that we can directly compare to the experimental arm, and that's probably a barrier for many programs. So in this case, we had a large over 1,000 patient patient level data set. The other aspect is that in an external control, we want to be able, there's always going to be unknown unknowns that we cannot control for. But as many of the known prognostic factors that are collected are vital to have within that data set. And the reason why clinical trial data is sort of naturally better, is that as part of a clinical trial, as I think everybody knows, is there's a huge collection of all of those, demographic data that may impact outcomes, age, sex, as well as baseline characteristics of the cancer, of the tumor itself, that we know for all cancers, that affect prognosis. So, MYC amplification for neuroblastoma, which was an exact match between the two data sets for the DFMO application, because it's such an important prognostic factor. So in this case, we knew that we had a patient level clinical trial data set where that information was collected systematically through the Children's Oncology Group when they ran, AMBL 0032. And thus we were able to do a propensity score matched analysis where we had 11 clinical characteristics that had been collected as part of AMBL 0032 and also as part of the single arm trial in 3B. And so that led to a very uniquely similar data set that is often, not possible to replicate in external controlled data sources from real world evidence, just given that there's often a much higher level of missingness on some of those important prognostic factors. So all of this requires sort of a lot of meetings with us to actually look at the external control data set and say, these are the factors that we and experts in the field think are very important to be matched to the experimental arm. And do you have these, and how much are missing? Because all of that sort of chips away at the certainty and the results, the more things that are missing.
So you did select patients out of that full data set as your comparative group. When I look at Alice Yu's paper from the study in 2021, the event-free survival curves are worse than the control arm that you guys selected, or the control set. And I guess I didn't dig too deep. Maybe it's in your paper, but what's the explanation for that difference? Because if you're selecting a population that may be a little bit better as your control arm, it makes me wonder, well, is the population that was done in the study for DFMO a better population than the overall population? Does that make sense?
Yeah. So, we tried to use the most sort of relevant and up-to-date data set. We were basing our EF, or the single arm trial based their sort of EFS assumptions on the EFS that was observed in ANBL 0032 in patients who had received the full battery of upfront therapy, including immunotherapy. And so that's and so what we sort of did, or what we were able to do, is because the vast majority of the 3B patients, the 3B is the DFMO study, had been on ANBL 0032, we felt that was sort of that really they were very much the same, the two populations, in that the only difference was that the remainder of the patients in the control arm had not gone on to join study 3B. And there's a lot of discussion that we had about sort of like what are the unknown factors that may lead a patient, to join another trial that may affect, prognosis. And that's discussed in our, that was some of the social determinants of health assessments that we did. But so, Tim, we sort of wanted to use the most similar population, and we had the AMBL 0032 data, and we knew that also that had led to the, approval of dinutuximab. So we were sort of using those EFS results as the comparator, because those were the patients from which, the study 3B patients were taken.
Well, and I did appreciate in the paper, you guys said, oh, what if these other factors were making things worse, and made some assumptions? Still, the analyses came out favorable for DFMO. So it did provide reassurance, I think, that, hey, this is really believable and important. And
yeah, and I think, our statistical teams and our statistical and epidemiologic teams that were, on our primary review team did an exhaustive search for anything that we could find in the literature that we could then that we could sort of put the results through. And as you said, we actually assumed the worst case, like plus some extra scenarios for for the experimental arm, in that, we sort of took the baseline poverty level and tripled it. Imagine that it would be potentially tripled in the control arm, to really bring the results through the most rigorous sensitivity analyses that we could, to see if the treatment effect held up. There's always uncertainty. There is uncertainty in the results, but we then had to make an ultimate determination on benefit risk, knowing that in this high unmet need, rare patient population, how much uncertainty is appropriate in a drug approval. Looking at the, sort of, with that being the risk, looking at the benefit, which in our assessment was a large treatment effect that held up to all of the sensitivity analyses that we put it through.
And Nicole, I'm going to go back to something that you said, is that this sort of approval process for DFMO this is the first time this has ever been done, leading to an approval for a product in pediatric oncology space. My sense is that that it was more because of some of the exceptional circumstances around this and the availability of a data set, that this was feasible, and that this type of process is going to be difficult for a lot of other drugs or products. And you've alluded to some of those challenges. Given the fact that there are a lot of single arm studies done in pediatric oncology, how do you think, and as you said, there are guidances on this that have been published by the FDA, but also with regards to now this published paper where it's very clear what the process was that led to the DFMO approval. Are there key sort of lessons learned there for when we look at the pediatric oncology landscape and the number of single arm trials that do exist, and thinking about setting ourselves up for success in the future?
Yeah, so that's a great question, and I have so many things to say about that. So the first thing is, I think that I think that when anyone is talking to FDA about designing trials, the ultimate goal is for there to be a labeling update. And some people may think like, well, who cares if there's an indication in the label, as long as we have a study, that shows that a drug is better, kids will start taking that drug, and all of our goal is to improve outcomes for children with cancer. So, but that's obviously the first and overarching most important goal, whether or not things get into a label or not. I would say that having an indication in a drug label is a benefit in and of itself, because of the way just that the healthcare system is set up in the United States, whereby if there's a labeled indication, it's typically something that would be covered by insurance and will become standard of care. So that's the sort of practical practical benefit to having something in a label that says this drug is indicated for the treatment of these children with this type of cancer. And then, so that's a practical benefit. Then the other, benefit is that if something has been sort of rigorously evaluated by a neutral, agency, us, the FDA, there can be sort of, I think, confidence from us all, in academics and at the FDA that is in fact, there has been demonstrated safety and effectiveness of this product for this particular indication. And it creates a sort of studied and vetted baseline for all future drug development to be measured against. So I would say that, so you're right, there are lots of single arm trials in pediatric oncology, and that in many cases, that's fine. We can make a marketing application based on a single arm trial. It really just depends on the endpoint, the effect of the drug, the magnitude of the effect, and it's all very, drug development program dependent. And so that's why we sort of encourage, or always, all of our talks always say, please meet with us early and often so we can discuss. For example, in the DFMO application, one of the unique things about it was that it was in the maintenance setting, so we couldn't rely upon response rate, which is our most, sort of common single arm endpoint. And there have been examples of multiple approvals, traditional approvals, that have been based on single arm data sets with the response rate. There are obviously nuances to that, but if it's a pediatric oncology indication for which you anticipate a response rate, well, maybe the single arm trial is enough. And that's why, that's why I think we want to be involved earlier, because maybe there's a perception that, there needs to be a single arm trial, and then that needs to go into a randomized trial. And it's very difficult to do randomized trials for a lot of different reasons. And thus the drug development program sort of doesn't, fails before it even starts. But I think that we are very willing to entertain flexible ideas for rare diseases such as pediatric cancers, for which there's an extremely high unmet need, and can often and have worked with small data sets and single arm data sets. So I think that those are all entirely possible to be successful drug development programs, with success defined by, goes into a marketing application and ends up in a label. Obviously, that's not to take away from the many successes that you all have had, treating patients on academic studies that have moved forward the standard of care for lots of different cancers.
Those are all great points, and I'm sure we could talk about this for a long time. We're over our normal time, but that's okay. I guess I think you talked a little bit about why this is a historic decision because of those. Is there anything else, sort of final comments you want to say about it before we close out?
Yeah. So you're right. It was a historic and exciting, it was an exciting approval for us. And it was a very unique approval. So I think Brenda said it very rightly, that it will be difficult for other drugs to utilize the same example, because there was a number of unique circumstances that led to the success of that marketing application. We're delighted to have approved a drug in this setting, but I think that generally speaking, we would like to be involved earlier with drug development programs so that we can make the most, we can minimize the amount of uncertainty in drug development as best as we can, still being willing to entertain flexible and creative ideas in patient populations that are difficult to study to, move the field forward.
It gives us all hope, I think, in pediatric oncology, that we'll be able to continue to make progress and get drugs approved for these rare diseases. And we appreciate all your work in this effort, and appreciate your coming on and sharing some of the inside scoop with us.
Yeah, thanks so much. And thanks so much for asking me. I really do appreciate being on this and being able to work with you and speak with you, because I think that just, furthers our partnerships, since we all have the same goals.
Absolutely. And thank you, Brenda, for co-hosting.
Thank you, Tim, and thank you, Nicole. This was an absolute pleasure, and appreciate so much the really important work that you do. So thank you, and your colleagues at the FDA. Thank you.
Thank you.
And thanks to the team at Solving Kids Cancer, a nonprofit charity dedicated to improving survival through creating novel treatment options for children. Remember, the more we learn, communicate, share ideas, and work together, the faster we'll reach the day when all childhood cancer is preventable or curable. As always, keep up the fight, and thanks for listening to This Week in Pediatric Oncology. And that's a wrap.
We welcome your comments, questions, or thoughts on topics for future episodes. Just drop us a note at TWIPO@solvingkidscancer.org. You can follow Dr. Cripe on Twitter at KidsOncDoc, send an email to Dr. Weigel at weige007@umn.edu, and find all TWIPO episodes at solvingkidscancer.org.